Good morning, everyone. Can you all hear me okay? No? Oh, okay. Can you all hear me okay? I don't have any conflicts of interest related to this talk. In other words, nobody's paying me to be here today. Okay. And I'm going to cover a topic that is very dear to me, as I imagine it is dear to all of you, which is brain reserve. I'll touch upon what we mean by brain reserve, and can cognition or brain reserve be enhanced? And if it can be enhanced, will that translate into meaningful functional improvement? And then finally, what can you practically do as individuals to help your brain reserve? Now, if you're fortunate enough to be Ben Bernanke, and your country is a little bit short of dollars, you can just print some, okay? Unfortunately, most of us are not that fortunate. And so we typically work for many years, in fact decades, to develop a nest egg and end up with a retirement amount, after which we might stop working, and then we start spending down that amount. Now, while we're spending it down, hopefully it's in some investments, and even though we're spending it down, some of it is also regenerating some income. And this is a graph, it's an outdated graph, but it kind of shows the idea that if you speed up the percentage or the amount of money that you withdraw from your retirement, the faster or the more you withdraw from your retirement, the faster it runs out. And the less you withdraw from it, the slower it runs out, and so it may stay with you for the duration of your life, given that you withdraw some and some of it is replenished. The thing that is shown on here is that, so if you withdraw about 4%, and some people now argue it should be 2.5%, but if you withdraw a certain small amount of your retirement, that your retirement can then last you for the rest of your lifetime, which is about 30 years after retirement. If you withdraw more or double that amount, you're going to run through it pretty fast. Now, let's say, so let's say you started with $1 million and you withdraw 4%, that's $40,000 a year. If you withdraw 8%, that's $80,000 a year, and that will run out pretty quickly. Now, if you started off with twice the amount, in other words, instead of $1 million, you start with $2 million, and you're still drawing $80,000. $80,000, it becomes only 4%, and so if you start off with a higher amount, you're able to sustain your retirement funds for a good bit longer. Now, this talk is not about money, and I would rather preserve my brain cells anytime rather than preserve my dollars. I think most of you would agree with me on that, and I'll come to this point towards the end of the lecture. But there is a certain parallel between money and retirement, as an example we're all familiar with, and what happens to our brain cells. This is why I'm showing you this slide. So this is not the Federal Reserve, it's called brain reserve. Okay, it's what do we start with? Do we have more nerve cells? Do we have more volume in the nerve cells that we have, more synapses? And then how does that protect us from everyday damage that occurs to our brains and allows us to still function? Now, cognitive brain reserve refers to the number of brain cells and the volume. Cognitive reserve refers to how well we function with the same amount of brain cells. So if somebody has fewer brain cells, but they're functioning at a particular level, and somebody has more brain cells and they're functioning at the same level, there's a discrepancy there. How come? That's one of the things we're going to talk about. So how did these concepts come about, brain reserve and cognitive reserve? Well, from the moment we are born, in fact, from the moment we are still a fetus in our mom's womb, we're losing brain cells. We're creating brain cells and we're losing brain cells. There's a very important reason for that. And I have a slide that explains that a little bit better. But basically, during development of the nervous system, there's a loss of a lot of cells. But that loss is part of what's called programmed cell death. In other words, we're built in a way where we have excess, and the excess allows us to form the connections we want to form, and then lose out the connections we don't need, that become surplus. It's a little bit like saying, well, if I keep collecting stuff at home, I won't have any more room in the house. But if I collect the stuff that I need and throw away the junk mail and the stuff that I don't need, I'll be okay. All right? So what happens in real life, and this happens across different animals and in humans as well, is that you do lose some cells. For example, if you had full webbing, which we don't really need between the fingers, that would be more of a hindrance for us than a help. But as we develop in the womb, that webbing is lost. Similarly, other animals have similar things happening where you lose some cells in order to remain with the cells that have the function that you want. So that's all part of programmed cell death. Now, during the intrauterine period of life, we also lose excess neurons. And somebody has taken the trouble to calculate that. Okay, 50,000 cells during each second. That's a lot of cells. Okay. What is really interesting for me as a brain scientist is that at least half of our entire genome is devoted to producing the brain. So the brain is very highly valued genetically. And in fact, also in life, 20% of the calories we consume are consumed by the brain. Even when we're sitting down and just watching or attending this lecture, 20% of the calories you consume are consumed by the brain. Okay. So as we age, we lose brain cells. Now, we can say, oh my God, I'm losing brain cells, but you've been losing brain cells all your life, and you're doing okay, all right? Goes along with memory loss, and that goes along with diseases like Alzheimer's disease, frontotemporal dementia, vascular dementia, and other conditions. So it's not just trivia, it actually has practical significance for us. This kind of information has practical significance for us as we see patients in the clinic. So one of the diseases that relate to brain reserve, because we lose so much brain reserve with it, is Alzheimer's disease. Alzheimer's is the most common dementia of the elderly. It's responsible for 60 to 70% of cases of dementia. There are over 80 causes of dementia, and Alzheimer's is responsible for 60 to 70% of them. Okay, so that's why a lot of times when you hear about dementia, you also hear about Alzheimer's disease or vice versa. But Alzheimer's is not the only cause. And so part of my job and the job of Dr. Foster and Dr. King at the clinic here at the university is for us to figure out, is it Alzheimer's? Is it something else? Now, if we were betting people, the odds are with us. We'd be right 60 to 70% of the time, even if we just said, hello, I think, okay. You have Alzheimer's disease. But, you know, if you happen to be the individual that's within the 30 to 40%, you wouldn't be very happy. So it's important to figure out what the actual diagnosis is, and then ultimately that will translate into treatment and advice that we give. Now, patients with Alzheimer's disease have distinct pathology. Pretty much anybody with Alzheimer's disease is going to have plaques and tangles. Plaques are abnormal protein deposits in the brain, and tangles are filaments that are formed within the nerve cells. So this is a picture of a plaque, okay, and it just shows a jumble in there. Yes, what you're seeing is a jumble, and we see these plaques. Now, in Alzheimer's disease, depending on the stage of the disease and depending on the severity of the disease, you'll also have these tangles. They're basically like black threads. In this case, they're so matted together that they just show as a jumble, but sometimes they look like braids under the microscope. Okay, it turns out that in Alzheimer's disease, you have to have both of these things, the plaques and tangles. Interestingly, if you have lots of plaques, you may or may not have dementia. If you have lots of tangles, you pretty much have dementia. But there are exceptions, as with everything in life. So there are a bunch of studies where people looked at individuals they had been following year after year for a while, who were normal, who functioned normally on their pen and paper testing. And any one of you who is familiar with our pen and paper testing, it's enough to cause anyone to tear their hair out. You know, it's an exercise in endurance because it takes 3 to 4 hours. And then we always ask you these questions that nobody can answer to test your limits. So it's so hard. And so if a patient has Alzheimer's disease, it's that much harder for them and that much more frustrating. But it's the best way we can figure out the extent of the disease and if it is Alzheimer's or something else. It's a tool that is very useful for us. So there have been a number of studies, and I've just put a sampling in here, where people looked at persons who had normal functioning on this rather strenuous test, and then died and donated their brains, and the scientists looked at their brains under the microscope and found, lo and behold, a lot of plaques and sometimes tangles also. But these are people who are functioning normally because they were being tested all the time. So go figure. So what is it? Why is it that the characteristic pathology of Alzheimer's disease can occur in one's brain without a dementia showing up? So these people are special in some way in that they don't show signs of dementia at the time that either at the time of death or nowadays we can measure the amyloid plaques in real life in vivo, and we also find that there are people who have these plaques but yet function normally. So it's possible that in those people, it's just that this is a precursor to developing dementia. But as I just told you, some people died and did not have dementia, even though they had these plaques. So it's not the final word, it's not definitive. So we have to figure out how can we be more like these people rather than the people who develop the plaques and tangles and develop dementia. So let me just, I mentioned a second ago that we can now measure the plaques in real life. And here's one study that uses a marker, a PET marker, that's positron emission tomography. It's a test that gives us a picture of the brain and gives us a picture of how much amyloid plaques there are. In this first image on the top line, you don't really see much uptake. In other words, you don't see much yellow and red. And after the person died, when they looked under the microscope, they did not find much plaques. You see, you don't have any of these blotches. In this person, they had a lot of uptake, okay? A lot of uptake, a lot of yellow, orange, and red. And on the scan, see, all of you can see these grains all over the place, and there's a lot of them. And the middle image is an intermediate image, and somebody who has some, but not as bad. Okay, but it's also obvious that they do have pathology on the scan as well as on the brain under the microscope. So we have a way nowadays of measuring if somebody has amyloid plaques in the brain. But remember, do these people have Alzheimer's disease if they have plaque in the brain? Maybe yes, maybe no. Just by looking at the scan, you cannot tell. It's very important to know that. So I've had some people bring me a scan. Doctor, my doctor said I have dementia because the scan is positive. Okay, well, let's test you. Do you have memory problems? No. Let's test you. They do this 3 to 4 hour test and they don't have memory problems or other cognitive impairments. So do they have dementia or no? They don't have dementia. They have a scan that shows pathology of Alzheimer's disease. So it's very important to know what we're dealing with. Now, it's possible that because they have pathology of Alzheimer's, they are going to develop Alzheimer's. But are they going to get it in 6 months, or 6 years, or 16 years? We don't know. Okay, we're still trying to figure those things out. We have some ways of trying to figure some of it out, but it's still not quite as fine-tuned as we'd like. Okay, so I know this is a busy slide, but the take-home message is that about 20 to 30% of normal elderly people have amyloid pathology on the brain if you looked at their brain after death or if you did the amyloid scans. And these are people who are functioning normally, so 20 or to 30%. It depends on the age group. The higher the age group, the higher the percentage. So, why is there a discrepancy? Why is it that some people have these amyloid plaques, have Alzheimer's disease, some of them have what's called mild cognitive impairment, which is when they have some mild memory loss and they're not normal because of the memory loss, but they don't have dementia because they can still function? Or why is it that up to 30 or 35% are normal? So, some of that could be because you need to have tau, the neurofibrillary tangles, also in order to show signs of Alzheimer's disease, and this is a prevailing hypothesis. But as we saw, sometimes people who also have tau function normally. So there are hypothesized to be different reasons for this brain reserve where somebody has amyloid plaques, but yet they're still functioning within normal. Some of it could be they have good genes. Some of it could be because they have a good education or had a lot of accomplishments in their life. So let's say somebody was a high school dropout, but they went ahead and developed a company that's thriving and very successful. So that's at least equivalent to a high education. Accomplishments in their life, so that's what I was referring to. And then it depends on the environment they were brought up in. Is it a rich environment? Is it a cognitively stimulating environment? Is it a socially stimulating environment? What about their nutrition? Were they eating healthy or were they eating junk food? And then do they exercise and have a healthy lifestyle or not? So I'm going to talk a bit about each of these because that's relevant to brain reserve. Okay. So one of the genetic risk factors for Alzheimer's disease is a gene called apolipoprotein E. If a person happens to have the E4 allele, one or both, they are more predisposed to developing Alzheimer's disease at an earlier age than if they didn't have that gene. it turns out that exercise has been associated with less cortical amyloid deposition in cognitively normal adults. So a person who's cognitively normal who exercises has less plaques than somebody who doesn't exercise. So this slide kind of shows us that difference. Over here, these are the people who don't have the E4 allele. This is the PET scan. It's like the PET scan that I showed you. It has a marker for amyloid, and it tells us how much amyloid deposits there are. So the people with low exercise have way more amyloid deposits than the people who have high exercise. Okay? So exercise seems to protect us against these amyloid deposits. Also, if you look at the CSF markers, and the CSF markers that we typically look at are amyloid beta 42, which is a length of a molecule, basically, of amyloid, which is this abnormal protein that tends to deposit. And it turns out that in the CSF as well, you do see changes. Now, so the less amyloid, in this case, in the serum, the less amyloid in the serum, the better. But the less amyloid in the CSF, it's worse. Okay? So in here again, you see that there's a difference between people who exercise and people who don't exercise, except that it's not statistically significant, at least in the CSF, in this study. Okay. Education. Does education protect us? There have been a number of clinical pathological studies. What do I mean by clinical pathological? It means that you look at people clinically, you evaluate them, you do the different testing, and then you look under the microscope and see, is there a correlation? So that's what we mean by clinical pathological studies. Such studies suggest that the relation between quantitative measures of Alzheimer's disease pathology, that is, if somebody bothers to look and count or estimate the count of plaques or tangles they see under a high-power field in the microscope. And so this is how we stage Alzheimer's disease, is we say, well, there are over 100 per field, or there are 10 to 100, or there are less than 10, for example. Okay, so that seems, the amount of pathology, particularly the tangles, tends to correlate with cognition and the number of years of formal education. So here's an example of that. Okay? So if you look at global cognitive function, okay, so you start off with so much global cognitive function. Now, the more Alzheimer's disease pathology you have in the brain, so this way going down is more pathology, okay? So the more Alzheimer's disease pathology, the less well you do on your global cognitive function. So there is a correlation between the amount of Alzheimer's pathology and cognitive function. And this is looking at different subsets of pathology. So the neuritic plaques, if you have the plaques, remember I told you if you have the plaques, cognitive function can be still normal, okay? Whereas if you have the neurofibrillary tangles, the slopes go down. So the tangles are more associated with cognitive function. Now, what about a person's accomplishments in life? So not only education, but mentally demanding jobs seem to have a protective effect. So if you look at the amount of pathology in the brain for somebody who, let's say, we've got two people performing at the same level. One of them has a much higher education, than the other one. So let's say one has a college education and the other one has only elementary school education. And if you look at their brains and the amount of pathology, the one with the higher education has a lot more pathology for the same level of performance. It means that they have had more brain reserve, and so they've been able to function at a higher level per unit of pathology than the person with the lower education. Okay? So their education has had a protective effect on their functioning. Now, another aspect is the environment. How rich is the environment? If you live in an environment that's quite stimulating, where you have to think, where you can play, where you can interact socially, there have been a number of epidemiological studies that suggest that all these conditions help delay onset of Alzheimer's disease. So people looked at transgenic mice where they developed these mice that have the genes for Alzheimer's pathology. So now they are transgenic. They're mice, but mice that have a human disease that was put into their genome. Okay? And so these are mice that are prone to, when they die or when they're sacrificed, if you look in their brain, you're going to see Alzheimer's disease pathology. So they took a batch of the same mice, siblings, okay, and put some in a rich environment and some in, just in a cage. And the ones in the rich environment and the ones in a cage were later sacrificed, and they looked at their brains under the microscope. It turns out that the ones that had the rich environment had fewer plaques than the ones that just had the regular cage. So that is very strong evidence, at least in mice, that an enriched environment helps protect. Doing such a study in humans would be extremely difficult. We don't exactly want to sacrifice them to look at their brains under the microscope. But fortunately, as I said, nowadays we can start thinking about such experiments because we can do the PET tracer or the lumbar puncture for biomarkers and be able to figure it out. And so far, the preliminary studies suggest that yes, an enriched environment does protect against some of the pathology of Alzheimer's disease. So, how come? You might ask. Well, it seems that an enriched environment results in a number of changes in us that are beneficial changes. One of them is that there is less cerebral inhibition. That is, the brain tends to be more active. There is enhanced neurotrophin expression. What does that mean in real English? It means that stimulating a person living in a stimulating environment, what that happens is it tends to result in stimulation of innate fertilizer, if you will, for the brain, of nerve growth factors, of different factors that the body produces that help nerve cells grow and stay healthy. Okay? So the enriched environment seems to promote those kinds of trophic factors. Now, you all know that if you lift weights, okay, it's going to stimulate growth of your muscles. All right? That we're familiar with. But it turns out also that an enriched environment has a similar or equivalent effect to brain cells. You won't develop a Popeye brain, but you will have more connections and healthier cells. Finally, it seems that there are changes that occur to how cells divide and how healthy those divisions are, so that you end up with more dendritic sprouts, that is, you end up with more connections between nerve cells. It stimulates the growth of these connections. Think of it as you have a tree and you just let it grow, and then you have another tree and you're giving the other tree fertilizer. Well, you can imagine that the tree with the fertilizer is going to grow a little bit faster and have maybe bigger fruit and more branches, right? A similar thing happens in our brains. This is another study that shows how social network, we live in an age of social networks, okay? Social networks, the more social networks we have, the more protective it seems to be. So basically just look at the slopes of these. In people who have high social networks, their cognition tends to be better than people with lower social networks. And people with higher social networks tend to have lesser neurofibrillary tangles than people with less social networks. And remember, the neurofibrillary tangles were associated with memory loss. So it seems that living in an enriched environment seems to also protect us, as well as a socially enriched environment seems to protect us. Okay, what about nutrition? Well, there's a lot of debate with nutrition. Part of the problem is it depends on what you've been eating all your life, and that usually changes over time. And we still haven't quite figured out, is it what you've been eating in school, or in midlife, or in late life? It turns out more likely earlier rather than later in life. Now, so you can always blame your parents. Okay. Anyway, so I come from Lebanon. Lebanon is a Mediterranean country. We eat a Mediterranean diet, so I always like this slide. Because a Mediterranean diet in several studies has been shown to have beneficial effects. Fruits and vegetables are protective, and reducing trans fats and reducing sweets seems to be protective. There's a very interesting study that was published in a prominent journal called Nature just a couple of days ago that showed that feeding mice 25% of their calories from sugar, just really non-nutritious sugar, resulted in either earlier death or poorer social functioning. Isn't that interesting? So it seems that the evidence is mounting that empty calories are not particularly good for you. Okay, and then certain fats are not good for you, but some fats, like the fats from fish skin or nuts, seem to be protective. But we have a lot to learn still from all of this, because you always find a study that says, oh yeah, I want my omega-3 fatty acids, I'm going to pop those pills, and then some other study comes and says, well, we really didn't see that association. But I would say that the odds are, if you eat healthy, that's going to be in your favor. Okay. In a very interesting study, for the people who love French food or cheese, okay, we did a study back in 2007 where we looked at cheese consumption and cognitive function in a group of over 1,000 community-dwelling elderly. And there was a dose-response effect in that the more cheese a person consumed per week, the less cognitive impairment they had. Okay. So I'm not saying go to a diet of cheese, but cheese in moderation may actually have some beneficial effect, either from the nutrients, the micronutrients in it, some of the amino acids in it, or certain essential fats in it. But this study really needs replication. I would love for somebody else to come and actually do a study where they, either confirm or challenge our findings. But again, I would get back to a balanced diet that is a healthy diet is probably going to be in your favor. Okay, what about exercise? Speed of thought and sharpness of thought have been associated with increased exercise. Cognitive performance on tests has been associated with increased exercise. In fact, when people have looked at MRI scans of patients who exercise more versus patients who exercise less, they found the thickness of the gray matter, that's the nerve cells, is thicker in people who exercise than in people who don't, suggesting that they have more brain reserve, more brain function. And aerobic fitness has been associated with, you remember that 3 to 4 hour test I was telling you about? Better performance on similar tests. Okay, so that's the word salad or letters here, AVLT, CFT, and PESA. They're just different kinds of tests that we do. And even in people who have the apolipoprotein E4, they perform better if they exercise. So it seems that that you can do something even if your genes tell you you have this predisposition, you can still do something to help yourself. Okay, so now, one of the things we look at is brain volume, or volume of the hippocampus, because that's really important. The hippocampus is very important for memory, and the hippocampus is strongly affected in Alzheimer's disease. So people looked at high exercise, individuals who do high exercise on a couple of different kinds of tests, exercises, versus people who don't do much exercise. And they found that the volume of their anterior hippocampus shrunk in the people who don't exercise. They normally do shrink, okay? But for people who did exercise, they got an amazing response. Now, there is no drug, current or imaginable, that's going to produce this, okay? Just so you know. I mean, I get sedentary, obese people who are obviously sedentary, okay, who are obviously not eating healthy, come in and say, well, my mom has Alzheimer's disease, you just diagnosed her. Do you have a pill for me? Yes, I have a pill for you. It's called exercise. Okay, so actually I have a prescription, and the prescription is exercise. It's not a pill. Okay. Now, they did another study where they had a group that did walking and a group that did more passive exercise, or flexibility and toning. Even though flexibility and toning seems to be of some help, the people who did the walking exercise had a lot more connectivity or functioning, communication between brain cells in different parts of the brain. And so the orange here shows you the increase in communication between different parts of the brain, the different networks. The brain has networks, okay? It's a little bit like airport hubs, okay, or airline hubs. So there are different areas that communicate with other areas on a regular basis. And communication between these areas increased in all of the people who had the vigorous walking. But it also increased in some of the people who had tone exercises, suggesting that exercise, no matter which, is helpful, but more aerobic exercise is probably more helpful for you. okay, and more healthy for the brain. We're very fortunate today because we can measure brain volume in vivo. We don't have to wait for people to die to be able, like 20 years ago, to be able to, look under the microscope. We're able to measure these networks and the functioning, and we're able to measure how active brain cells are, and we're able to measure the Alzheimer's pathology as it deposits. And so over the next 10, 20 years, you're going to see a tremendous amount of studies that look at all these things that we've learned from epidemiology and try and develop experiments to determine, well, can we show cause and effect? We've shown association through the epidemiological studies, but association does not equal causation. Back there, here, and then there. Yes.
What about intervention? I'm an educator of behavioral skills. Occasionally I do have Alzheimer's plan, and I'm really at a loss what to do.
Okay. Yeah. So the question is about intervention rather than prevention. And so for intervention, we recommend everything I've told you about, but it may be a little bit too late. Nonetheless, it's not so too late to have a better quality of life, and all of these measures do provide a quality of life. I would like to introduce Liz Garcia, okay, and she is our health educator. And we advise every patient, actually we prescribe and we give an education prescription to every patient to meet with Liz so that she can talk to them about these interventions. So maybe you can catch Liz afterwards about the intervention part.
Is there any neurogenesis at all?
So the neurogenesis, it sounds like there is some, but I don't think it's anywhere near enough to help compensate. So people are looking at strategies to promote neurogenesis or what might come 20, 30 years down the road with stem cells and that kind of thing. Okay, and then there was here, okay, afterwards. Yeah.
Would you please describe or illustrate for us social networking?
Okay,
because we
so
have these media devices for so doing. Do your studies differentiate yet?
Okay, so what I would say about the social networks is any network of friends, regardless of how you keep in touch with them, in person, by phone, by email, over these internet media, whichever, all of it is good, okay? This is not a plug for simply the internet social networks. No. Okay, this is any, your neighbor, okay, or your church group, or your social group that you go out with to see to the movies or dinner. Okay. Yes.
I was going to ask you about vigorous exercise. What do you consider vigorous exercise, and would just walking maybe 3 miles a day?
So walking, yes. So vigorous exercise, define vigorous exercise. Anything that will make your heart rate faster and your breathing harder. Now, a lot of us cannot exercise 30 to 45 minutes straight, but we can do it in 10-minute increments or 15-minute increments. Ramping up time and settling down time does not count. It's the time where, yes, I'm afraid so, where the heart is pumping faster and the breathing is harder. And do it at your capacity, or do it, in consultation with your doctor. One question here and then another one there. Yes. So the hearing loss and dementia is an interesting question because you can have impairment of comprehension with dementia. And a lot of us, as we age, we lose our hearing. A lot of us, as we age, develop dementia. Is there a causation or is it simply an association? It's a hot topic of investigation right now. But it's worthwhile checking hearing if you're concerned about cognition. Yes?
Do you have anything, if there's like a memory loss or brain function from like concussion from something right away instead of long term?
So there are a number of cognitive rehab programs for people with head injury. And it gets a little bit more complicated because there are certain medications that are supposed to help preserve the brain immediately after the injury, and then there is a very structured rehabilitation depending on where that injury is. So it becomes it's a little bit off topic for the brain reserve when it comes to Alzheimer's disease, but it does have to do with brain reserve also because some people for the same injury seem to do better than others. Okay, do we have, okay, 2 more questions. All right, so one here and then one there, and if you want, just catch me afterwards. Okay, so, yes.
Do all of these suggestions relate to the non-Alzheimer kinds of dementia as well?
So certainly they relate to vascular dementia, hardening of the arteries. It's unclear how much they might relate to Parkinson's or Lewy body disease or frontotemporal dementia, but they seem to make enough sense, and we see improved quality of life in a lot of our patients who lead more active lives that we recommend it across the board. And last question, and then I'll get to you. I think. Yeah.
You answered my question.
Okay. Yes.
How do you participate in any of your studies?
Contact us at utahmemory.org. And I promise you, she was not a plant. Thank you all very much. I'll be outside for a few minutes.